Ciao. Hello girls, it's Mrs. Metcalf here again, and this lesson is going to be about passive transport, but a different type to the last sort of passive transport. This one is called osmosis. So just to put it into perspective, we began by learning about the cell membrane and the structure of it, and we learnt that substances have to pass in and out of the cell membrane. The cell membrane controls what passes in and out of it because it has a selectively permeable or semi-permeable membrane. So we've talked about the first type of passive transport, which was diffusion, and now we are moving on to the second type of passive transport, which is osmosis. And really all osmosis is the diffusion of water. Are you ready? So, by the end of this lesson, you should be able to describe what osmosis is and explain why it is important in living things. And amongst other many living things that osmosis is important in, it's particularly important in fish. And I thought this is a pretty cute little fellow. So, what is the definition for osmosis? Osmosis is the spontaneous net movement of water molecules across a semi-permeable membrane from areas of low solute concentration to areas of high solute concentration. So let's break that down a little bit and review what these words mean. So spontaneous means no energy is required. Net movement means the overall movement. So some molecules will be moving one way, other molecules will be moving another way in the opposite direction across the semi-permeable membrane. This is when you take away, it's the total movement of the molecules moving to and from a cell, I guess. We know what a semi-permeable membrane is, which means only small molecules can pass in and out by diffusion. Now, solute, that is the kind of the opposite to solvent. So water is the world's most common solvent, and solutes are what we put into the solvent. So it might be sugar, it might be salt, for example. And the next thing you need to know is that osmosis acts to equalize the concentration of the solute, so for example the sugar or the salt, on the two sides of a semi-permeable membrane. So you can see here we've got some sort of a test tube type setup, a U-shaped test tube. In the middle there is a semi-permeable membrane, which means it will allow some small particles to pass through. Now on the right hand side we've got a concentrated solution, so it might be lots and lots of sugar particles, or it could be salt. If the right hand side is concentrated and the left hand side of the semi-permeable membrane is dilute, so you can see not very many sugar or salt particles, so the solute, versus the right hand side, the water is going to move by osmosis from the left hand side to the right hand side, and you can see that the level of the water has risen. Originally both levels of the water would have been equal. So remember, diffusion is the net movement of molecules from high to low concentration. So you can see down here, particles are moving in both directions. They're moving from left to right, right to left. In simple terms, osmosis is a special type of diffusion. Water moves from more dilute solutions, which have high water and low solute. Remember, solutes, for example, sugar or salt, to more concentrated solutions. So where you've got low water but lots of solute. So for example, lots of salt or lots of sugar. This happens across a semi-permeable membrane, such as the cell membrane, which is what we've been talking about. So this picture here shows, again, some sort of container with a semi-permeable or a selectively permeable membrane in the middle. So it allows only the small particles through, which in this case are water. The sugar particles, however, are too large to fit through the holes or the pores in the membrane. Because of osmosis, all right, the small water particles are going to pass from the left-hand side to the right-hand side where you have the high sugar concentration, and this will cause the water level to rise. So let's just review what is a semi-permeable membrane. A semi-permeable membrane is a type of biological membrane, which means membrane in living things or organisms, that will allow certain molecules or ions pass through it via a transport mechanism. So this could be, the transport mechanism could be diffusion, which we've already learnt about, osmosis, which we're learning about at the moment, facilitative diffusion, or active transport, and we'll learn about those two in the next two videos. One example of a semi-permeable membrane, I hope you know already, is the phospholipid bilayer, which makes up the cell membranes in almost all living organisms. And here's a diagram of that, of a cell membrane with the phospholipid bilayer, and you can see that these would be oxygen molecules easily diffusing through the cell membrane. Osmosis in living things. So water molecules are transported across cell membranes, both plants and animals, tonoplast membranes or vacuole, or the membranes around vacuoles, or protoplast membranes, only found in plants, by diffusing across the phospholipid bilayer or the cell membrane. Osmosis is the main way in which water is transported into and out of cells. So note, osmosis is water, the movement of water only, no other particle, just water. When a cell is submerged in water, the water will move into or out of the cell depending on whether the solute concentration outside the cell is higher or lower than the solute concentration inside the cytoplasm of the cell. Remembering the solute is what is dissolved in the solvent, which is water. This makes osmosis a vital process in biological systems. Tonicity. Tonicity refers to the relative solute concentrations of two environments separated by a semi-permeable membrane. For example, the cell membrane. The prefix hyper refers to more solute. For example, sugar or salt or ions. Iso refers to the same amount of solute, and hypo refers to less solute. So here we have a hypertonic solution. So there's lots of solute particles compared to water molecules. Iso, in the isotonic solution, you've got equal numbers of solute either side of the semi-permeable membrane. So that's a semi-permeable membrane. We've got 4 solute particles on the right-hand side and 4 on the left-hand side. Hypertonic means that you've got less solute. So you can see there's only 2 particles of solute here versus lots here, 6 or 8, and lots more water in the hypotonic solution. So what will happen if a cell is placed in one of these environments? The environment into which a cell is placed will determine whether it will gain water or lose water. When a cell is placed in fresh water, the concentration of solutes in the cell's cytoplasm is higher than the external environment, because fresh water will have no solutes in it, virtually. The cell will gain water under these conditions as water moves from an area of low solute concentration, that's outside the cell, to an area of high solute concentration via osmosis. So the water is going to move from outside the cell into the cell because there are more solute particles inside the cell than outside the cell. So 4 versus 3. And remember, this is greatly simplified. It's just to give you, paint the picture. In this situation, we say that the cell's external environment is hypotonic, relative to the cell's cytoplasm. Okay, moving along. If a cell is placed in salt water, the concentration of solutes in the cell cytoplasm will be lower than the cell's surrounding environment. Okay, so this might, for example, be the ocean. The cell will lose water under these conditions as water will move from the region with a lower solute concentration inside the cell to the region with a higher solute concentration outside the cell via osmosis. In this situation, we say that the cell's external environment is hypertonic relative to the cell's cytoplasm. So that means that outside the cell's external environment, there are more solute particles outside the cell than inside the cell. When a cell is placed in a solution which has the same concentration as the cell's cytoplasm, there is no osmotic gradient. In other words, inside equals outside, if you like. In this situation, water molecules will pass in and out of the cell at the same rate, and there will be no net change in the volume of water in the cell. In this situation, we say that the cell's external environment is isotonic relative to the cell's cytoplasm. So the number of solute particles inside the cell, 3, equals the number outside the cell, also 3. And again, this is greatly simplified, but just to give you the picture. When an animal cell is placed into a hypotonic solution, it will swell and burst. In red blood cells, this is called hemolysis. An animal cell will burst because it does not have a cell wall. This is why it is important to maintain a fairly constant water level in the blood. And this is part of a very important process that goes on in the human body in all sorts of ways called homeostasis. So basically, so in this case, in a hypertonic solution, there's a high concentration of solute particles outside the cell, so the water in the cells will go outside the cell, something like this, and the cells will shrivel. If the blood cells, red blood cells, are in an isotonic solution, then the blood cells will be their normal shape because the amount of water going into the cell is the same as the amount of water leaving the cell. If, however, red blood cells are put into, sorry about that, girls. So if red blood cells are put into a hypotonic solution, which means a low concentration of solute particles, for example, salt, water will, sorry girls, small interruption. So hypotonic solution, low concentration of solute outside the cell, high concentration of solute inside the cell, so the water will pass by osmosis out of the cell into the cell, as it's pictured here, and because red blood cells don't have a cell wall, they are likely to kaboom, burst. Okay, on the other hand, when a plant cell is placed in a hypotonic solution, it will gain water and expand to become turgid. They don't burst because of the cell wall. That provides support for the cell. So the cell wall acts to provide support and protection to the cell membrane as the cell gains water, preventing it from rupturing. This helps support the plant. Now, if a plant is in a hypertonic solution, that means that the amount of solute outside the cell is higher than inside. Water will leave the cell, and if that goes on for too long, the cell becomes plasmolyzed. And once a plant cell becomes plasmolyzed, that's kind of past the point of no return. That's when you forget to water your water plant for weeks and it doesn't come back. If, on the other hand, it's in an isotonic solution, there'll be water going in, there'll be water going out, the plant may be flaccid or a little bit limp. And what ideally you want is the plant to be in a hypotonic solution, which means low solute concentration outside the cell, high inside the cell, so the water will pass by osmosis into the vacuole and the cell will be turgid. That's when your plant looks nice and healthy. Okay, so this is what we've just been talking about. When an animal cell is placed in a hypertonic solution, the cell will shrink in a process called crenation. This decreases the available surface area, so this is why it's relevant to biology, and prevents the cell from functioning correctly. So it's, for example, it wouldn't be able to pick up oxygen, which is its main job. When a plant cell is placed in a solution which is hypertonic relative to the cytoplasm, water moves out of the cell. This causes it to shrink, become flaccid, and eventually the cell will become plasmolyzed. What does plasmolyzed mean? It means that the cell membrane separates from the cell wall due to lack of pressure of the vacuole against the cell wall. This causes the plant to wilt, and we've probably all seen that. So if your plant sort of starts to wilt, it's not too bad if it gets to this stage. If it gets to this stage, I think it's past the point of no return. Okay, so as cells are affected by fluctuating water levels, it is important to regulate the level of water in cells. The maintenance of fairly constant water levels is called osmoregulation. Organisms are adapted in different ways to achieve this. For example, ocean fish drink seawater. This would dehydrate their cells if it stayed in the body, so the fish has systems to pump out and remove excess salt ions. So you can imagine if there's lots of salt water in here, then the water is going to pass by osmosis from the fish cells out. And that would not be good for the fish because it would dehydrate their cells. So the fish has it does drink the seawater, but at the same time, it has these processes to get rid of excess salt ion. And that's called osmoregulation, keeping the amount of salt in the fish's body or the animal's body at a constant rate. Okay, so let's recap what we've just learnt.
Before we take a look at how osmosis and tonicity affect a cell, let's review what each of these terms means. Osmosis represents the diffusion of water across a semi-permeable membrane. The term tonicity refers to the relative solute concentration of two environments separated by a semi-permeable membrane. In other words, by comparing the tonicity of the solutions, you can determine the direction in which osmosis will occur. To demonstrate how tonicity affects a cell, let's place some red blood cells into a beaker containing pure water. In this case, the solute concentration is greater inside the cells than in the surrounding water. In other words, the contents of the cells are hypertonic in relation to the hypotonic contents of the beaker. Because of this, Osmotic pressure results in the diffusion of water across the membrane and into the cells. Over time, if enough water enters the cell membranes may burst. This is called lysis. Now let's place the red blood cells in a beaker containing a solution of salt, such as sodium chloride. Since the contents of the beaker are hypertonic in relation to the interior of the cells, the water within the cells will diffuse across the membrane and into the contents of the beaker. This causes crenation, or shrinking, of the cells. If the red blood cells are placed in a beaker whose contents match the tonicity within the cells, then there is no net gain or loss of water. The environments within the beaker and inside the cells are said to be isotonic, or the same. An important thing to remember is that osmotic pressure always causes water to move from a hypotonic environment toward a hypertonic environment.
Okay, so girls, let's wrap it up. So, in summary, hopefully you have now learnt to describe what osmosis is and explain the importance of osmosis in living things. The last thing I want you to do is I want you to copy this summary, which is basically everything that we've just been over. Thanks, girls.